Welcome students. This is for 5th semester B Pharmacy students. I have taken a question paper for our discussion as I told in the classroom. This is December 2023 question paper of KUHS University question paper. If the student have a practice of answering a university question paper, I feel it will be a good experience for you to how to write, and sometimes some of the questions will be differ from the notes you prepare or my lecture which I have given. So it will be interesting for you to write down once, and if you submit to me, I hope there is no difficulty for the students to get 50% of mark minimum, and even the students can score more than 95% and they can get rank in the subject, rank in the particular semester. So this is my aim. So I hope your cooperation is needed to get into this answering this question paper. I have given, actually you have 3 hours question paper, so I cannot give you in one lecture within an hour. So at least it needs, I made it into 3 parts, so such that I hope it will be easy for you to write down the assignment as I expect from you to listen and to write down the notes for the answers, actually for the university examination. So as I told, I have taken for pharmacognosy and phytochemistry 2 subject for the 5th semester B Pharmacy student. The university KUHS question paper December 2023 for the 5th semester B Pharmacy students. The code number, question code number according to the university 524006, and you will write your register number. I have taken the question paper. This is for degree supplementary and as well as regular also. December 2023. Cognosy, pharmacognosy and phytochemistry 2 according to the scheme 2017. The time is 3 hours, and marks which you are going to write is 75 marks. And you know very well the model. You have 2 essay questions, and you have 7 short notes and 10 brief 2-mark questions. So we will discuss all. So what is the answer all the questions to the point neatly and legibly. Do not leave any blank pages between answers, please. And indicate the question number correctly for the answers in the margin, outside the margin. In the margin space, don't write inside. You can write in the margin so that it will be easy for the evaluator to find out the question number and to give you the proper mark which you deserve it. So answer all parts in a single question together. See, for example, a question answer, if you are writing, don't write in 2 places. Question number 8 is from the page number 1, and question number 8 the same again repeatedly in page number 11. Like that, don't try to make a confusion for the evaluator. Write always a question in a single area. Leave sufficient space between answers. Draw label diagram, flow charts wherever necessary. It's very important points. And also in pharmacognosy, when you are drawing any microscopy, macroscopy diagrams, draw by pencil. Don't draw by pen. Use pencil charts. When you write lines, you bring small scale for writing examination. It's needed. It's allowed for you to bring your in examination hall. Pen, pencil, rubber, sharpener, these all are allowed. So you have to do it in that way. In essay question, they have asked in December 2023, 2 questions you have to write, 10 mark question. Number 1, explain the role of chromatographic techniques in isolation and estimation of herbal drugs. And question number 2, essay question, they have asked, define and classify with examples. With examples you have to write. Write the source and the chemicals, constituents, and the method of extraction of senna. So these 2 questions we are going to discuss what should be written and how to write. And also short notes, 7 short notes, 5 mark questions. Write a note on ligands and carotenoids. Actually ligands is in the unit 3 and carotenoids in unit 4. They have taken together in 5 mark question you have to explain. Explain tracer technique, 5 mark question. Describe the anatomy, that is anatomical structures, microscopy and even macroscopy if you want it, but usually microscopical details of digitalis leaf. And describe method of production and estimation of Taxol. Production and estimation of Taxol. And differentiate the 2 benzoins, Sumatra benzoin and semabenzoin. In resins, these two types of species and 2 types of the product in unorganized drug, how they are differentiated, 5 mark question. And describe the method of isolation and analysis of podophyllum, Indian podophyllum, how you can take podophyllum, how you are analyzing and how it is as isolated from the crude form. So 9th question, explain the industrial production and estimation of vincrestin and vinblastin from vinca plant, how you are industrial production, they are asking 5 mark question. So we will discuss this 5 mark question also. And answer briefly 2 mark question, 10 questions they have asked. Give any 5 application of spectroscopical methods like UV, IR, NMR, mass, IR, all these things. What is the applications? How it is applied for various analysis of herbal products and herbal drugs, and about phytochemicals, how you are using this spectral data or spectral instruments. So we will discuss about. They are asking 5. I have given some more details to have the students to write down, and so that it will be easy for you to write the exam. And number 11 question, 2 mark question, write the biological source, that means botanical source, chemical constituents and uses of. It's not clear for you to see. You can, I will just show you because it's for, I'm just pulling down, and so that it will be easy for the students to see the question paper clearly. You can see here now of mentha they are asking, okay, and this is the, we can see that they have asked uses of mentha. And the 12th question, what is supercritical fluid, SCF, with liquid carbon dioxide? What do you mean by this fluid, how it is utilized, how it is used for chromatographic technique, a new technique which I have been explained for you in separately. This is 2 mark question. You have to write what is this supercritical fluid, liquid carbon dioxide. If you write only simple word, they will not give you 2 mark. You have to write about its application, how the chromatography work, some details should be written. Morphology of Rauwolfia. You have to write the morphological character, not microscopical, morphological. So botanical source and as well as the morphological external structure. You should know it's a root and it is called a snake root or diagrams, and you should explain it. And write the botanical source or biological source, chemical constituents and uses of Asafoetida. Asafoetida, Kayam. You should know the botanical source, chemical constituent and uses. 2-mark question. And they have asked 2-mark question, write the application of gel electrophoresis. What do you mean by gel electrophoresis? What is its application? What is its application towards pharmacy or pharmacognosy or phytochemical investigations? How you are going for any type of structural elucidation, or what is the application for characterization of chemicals? And the 16th question, 2-mark question, name the flavonoids present in licorice. You should write the source of licorice, name of the licorice, and you have to write what type of flavonoid is present. Give their medicinal importance. What is its use in the pharmacy or medicinal use? And write in the 17th question, write the method of estimation of forskolin phytochemical and its a source and how you are estimating it from the crude drug, forskolin. So it's an interesting to write. If you know the answer, the student, once you listen, once you write, it will be helpful for you, these type of questions which they ask, even the 2-mark question. Define alkaloids, give examples and mention its general properties. So you see the question, you know alkaloids and you should define it and give some examples. At least you can write pseudo-alkaloids and proto-alkaloids examples and mention its general properties. Even chemical test also general properties and what is its basicity. All these things can be written in 2-mark question. And in 19th, the last question, 2-mark question, write the chemical test to differentiate pale catechu and black catechu. In tannins chapter, we have pale catechu and black catechu. In what way you are going to differentiate them? They are tannins, but still they both are having some constituent content differences, and as well as chemical test can be given for differentiating. So interesting questions which we have read. It's going to be discussed with you, and if you listen and then you write, I hope the student will have a good idea about a question paper, how I should write. So first question is, explain the role of chromatographic techniques in isolation and estimation of herbal drugs. They have asked role of chromatographic techniques. You studied many chromatographic techniques, the basic principle of chromatographic technique. So the students should be thinking that what I should write, whether I should write about TLC or paper chromatography or column chromatography or HPLC or HPTLC, or I have to write about gas chromatography or about the that is super critical fluid carbon dioxide chromatography. There are many. So what I should write, or gel permeation chromatography or ion exchange chromatography, you should mention every chromatography in a short manner, and you have to write the role, what is its role, what is its application, and you have to write some definitions. I am giving in that way so that the student will have an idea. I have explained individually all the chromatographies in a wider manner, but here all the chromatographies together they have asked, so the student should be able to write some of the points as I have shown here. So here first you have to write chromatography is widely used for the separation and identification of compound of a mixture, or as I told in the class, it's a very simple, a student can write. Chromatography is a technique which is used to separate mixture of compounds. Chromatography is also very suitable for extraction of plant materials, especially when a small amount of material is available. When you have a very small amount, it's possible to extract, isolate when the component closely resembles each other, like alkaloids and anthraquinones. They are closely structurally related. You can go for chromatographic technique to isolate them or to separate them. In the latest form of chromatography was applied only to colored, earliest, not latest, earliest, that means the olden days, colored component they used to make in the compound, and we can differentiate. Now colorless also can be identified, as we have done in the lab. You might have done drag and drop reagent for detecting alkaloids, ninhydrin for detecting amino acids. You have to remember what you did for chromatography, like paper chromatography, thin layer chromatography, practical sense also the student should remember when you writing theory. So in the olden days forms, the chromatography was applied only to colored compounds, and resulting separation could be followed by visual observation. Previously, like this as shown in the colored one, even in the column or in the paper, colored component can be easily separated by means of their color. But then they found out, but anyhow the basic chromatography can be divided into three by means of their basis. On the basis of interaction with solute and stationary phase, we can have adsorption chromatography, partition chromatography, ion exchange chromatography, and size exclusion or molecular sieve chromatography. On the basis of chromatography bed shape, you can have 2-dimensional, 3-dimensional, and in 2-dimensional TLC and paper chromatography. In 3-dimensional, as I told, column chromatography, it is round and it has X and Y and Z axis. So that is an example for 3-dimensional chromatography, column chromatography. On the basis of physical state, we can have liquid partition chromatography applied, that can be called as liquid chromatography, and even solid and liquid sometimes you can have, that is even adsorption. Apart from that, gas and liquid also can be used in the chromatography, that is gas chromatography. So the basis of physical state also we can differentiate. So this is how the chromatography is basically, they can be classified. Apart from the techniques which you are studied and you know in the lab, I have, and also in the lecture separately which I have given for you, they are column chromatography techniques, paper chromatography, thin layer chromatography, high performance liquid chromatography, gas liquid chromatography GLC, and high performance thin layer chromatography HPTLC, superfluid chromatography SFC, and gas solid chromatography is also there. Not much, we are not using it much. It is solid and the gas can pass through in a silica gel columns is also possible. So anyhow, these all chromatographies are commonly which we know. So what is its application towards our pharmacognosy and phytochemical investigation or herbal product investigation? That's what the question they have asked. So first we have to write about column chromatography, which we know. In column chromatography, we are packing column as a stationary phase, maybe a solid or a liquid. If it is packed with a solid, then we will say the principle of adsorption. If it is packed with a liquid, then we can say it is a partition. Generally, the column chromatography is mostly based on the adsorption principle. Usually, we are using like a burette long tube with solid packing in adsorption column chromatography. The extract which are to be analyzed should be dissolved in the mobile phase and added in the top of the column, and then the individual component will move in different rates according to the affinity. Upon the affinity towards the stationary phase or the adsorbent which you are using, we can say that more affinity, which is very strongly affinity to the stationary phase, will be separated last, and those which are having less affinity or no affinity towards the stationary phase will move faster and separated first. So you can see this is the column as an example, and we can show that how a stationary phase is added in the top of the column like a burette, and how the separation, the colored separations can also be shown, and easily you can write about the elute solutions. And column chromatography techniques include conventional column chromatography like flash, and preparative HPLC also comes under a column because it's very small column, and is also HPLC also column. It's a column which is only, as I told in the class, it's only in very small columns. Even 10 centimeter columns are there, and 30 centimeter columns are also there, and are essential to isolate purification. What is its application? That's what the question asks. Purifying and quantifying bioactive compounds from the complex of herbal extracts. They enable the structural elucidation and the standardization of raw materials, quality control, and the identification of active constituents for drug development. These all can be done by a column chromatography. And thin layer chromatography, which we know about in the lab, which we do, and we can have the plates and a diagram of a developing chamber, and the TLC, is an adsorption chromatography. When you are using a solid thin layer like silica gel, stationary phase is a thin layer adsorbed and held on a suitable backing, usually a glass plate, and separation of the compound present in the plant extract depends on the difference in their adsorptive or desorptive behavior in respect to the stationary phase. So TLC involves in the layer of adsorbent mixed with the binder such as calcium sulfate, gypsum. So that's the stands for G, silica gel G, which spread on the glass plate in a thin layer and allowed to dry or activate. And the plant mix is applied as a spot near to the base of the plate, as we have done in the lab, which is then placed on a closed glass tank as shown in the picture containing the developing solvent. So its application, you can see they are asking about what is its application of TLC. So you can say alkaloids need usually an example. What you did, you can write moderately polar solvents, good separation, either ethanol 95:5 can be used, so that you can get this type of separation, and it is a good separation can be done. And if you see more polar solvents, suppose if you are using pure only methanol, when would be preferably adsorbed, the alkaloids would be carried along with the passage of the solvent. It may be carried with the solvent, resulting a poor separation as shown in the picture. You can see it will be carried along and the tailing will be there. You can see that is wrong separation. And you can see the next one, even it can be shown in the top also, that is also a wrong separation when it is more polar solvents like methanol if you are using instead of ether and ethanol combination, what I shown in the 95:5. And on the other hand, if you are non-polar solvents which you are using chlorocyclohexane, would be unable to move the alkaloids which is present in the spot which you have kept. So it may be display, it will not display the, it will not move the, it will not get separated, display the alkaloids which is in the adsorbent layer. They would be remain in the near to the baseline itself as shown in the picture. It may not move, you can see, that is also wrong. So the solvent system selection is very important. So you can write what solvent system you used in the quinine sulfate and your cinchona extraction, what you did in the lab. You could write that one, toluene, acetone, ethanol and ammonia, how you used as 45:45:7:3 ratio, how you used with the drag and draft reagent for detecting TLC can also be written. How you are doing application of an alkaloid can be easily. Additional factors of separation of alkaloids. If you using an aluminium thin layer instead of silica gel, neutral aluminium and neutral solvent should be used. So if you are using silica gel G, slightly acidic due to the method of preparation, the alkaline solvents such as you can use acetone, water, 25% ammonia like 90:7:3 make a good separation, that also can be used. So you should write this type of mobile solvent system, and if you explain about the problems which is good, which is bad about the techniques, it's interesting for the evaluator to give you the full mark. So separation of volatile oils by means of TLC also. Just an example, the constituents of volatile oil, mainly non-polar terpenes, are therefore best separated with corresponding non-polar solvents such as chloroform, benzene mixtures can be used. Certain oils may be more polar solvents for clove oil and phenolic components like eugenol. You can go for a little more polar solvents can be used. And separation of sugars, carbohydrate, sugars like mixtures. If you want to do this, pictures how a big slide, how they separated with the different colors of identification. This mixture produced by hydrolyzing starch or gums, and they are strongly polar, usually sugars, and therefore strongly adsorbed on the silica. As if silica is and alumina, they will get strongly adsorbed. Therefore, strong polar solvents are used to separation on these thin layers. They are not generally satisfactory, but resulting in obtain result is good result is obtained with weakly polar adsorbent such as cellulose. Even paper chromatography will be very much comfortable, or cellulose itself you can coat it without water cellulose coating, and polar solvents such as butanol, N-butanol, ethanol, water 5:4:1 can be used. Alternative paper chromatography should be used for this type of very polar components. Sugars can be more preferred. So this is what the student should know. TLC, thin layer chromatography, is compare better separation tool than a paper chromatography, and through the adsorbent term use various types of thin layer chromatography, but principle of separation may be any one of the four fundamental separation can be done in TLC when you are changing the stationary phase. Ion exchange chromatography by coating with resins, partition by means of cellulose with water or any impregnated solids with water linkage, and absorption chromatography normal, which we are thin layer silica or alumina coating. Molecular exclusion like dextrin or agarose molecular gel when you are coating in a glass plate you can follow. And principle of separation totally upon the coating substance. Stationary phase over the plate, applied over the plate, decides what type of principle you are following. In silica or alumina is applied as coating substance, adsorption principle take place. So if coating substance Sephardex, as I told, or agarose or dextrin, molecular exclusion will be the principle of separation. Ion exchange, as I told, resin with the maximum of resin which can be coated a substance, or a cellulose like coating substance. Adsorption, adapt partition with the water can also mechanism can be possible. So this is what and a thin layer chromatography is advantage form of HPTLC, high performance thin layer chromatography is available, which advanced one plays a fundamental role in phytochemical analysis, isolation and estimation of herbal drugs. So due to simplicity, low cost and high high throughput TLC is frequently used, that is HPTLC used in rapid screening and authentication and quality control of complex plant extract. It's an application. High performance thin layer chromatography is a powerful analytical technique which is wonderful suitable for qualitative and quantitative analysis tasks. This development of chromatographic fingerprints plays an important role in quality control of complex herbal medicines. So you see, it's a very interesting, even paper chromatography, you would write a small, take a 10 mark question, draw the diagram and how the descending technique, ascending technique, how is the paper, how it is hanged for a descending technique and ascending technique. So this can be written by you very easily, which you have done in the lab, showing the filter paper, how it is prepared, and plays a fundamental for phytochemical analysis, isolation, estimation and herbal drugs. Due to its simplicity and low cost and the effectiveness of separating polar hydrophilic compounds like amino acids, sugars and glycosides, while often used for qualitative identification, specialized techniques like two-dimensional paper chromatography allows for a separation of complex mixtures, while densitometer can be used for quantitative, enables quantitative estimation. So you can write the application, and also schematic diagram of HPLC if possible you can write, not explaining the whole thing. You can write the solvent, pump and injecting spot, guard column and the small column, sophisticated and minute, very interesting column of HPLC column and detector, different detector which is used like diode detector and UV detector, and we can have a collection of waste and it will be, detectors are connected with data computer. So this you can have an parts can be easily mentioned, and then you can go directly for the application. What is its application? Because our question is asked about what is the main different chromatographic technique applications and use in pharmacognosy. So you can see separation of constituents can be done, component and herbal extract, identification and quantification of very minute quantity compound, identify and measure the amount of compound in herbal mixtures, and estimate or establishing fingerprint profile of the benzene or aromatic compound, create a fingerprint, a standard or a marker, creating a profile for a chemical constituent mixture and a crude extract and comparing with that, and standardizing complex herbal drugs, estimates the presence of active chemical and biological markers. These all can be done. And gas chromatography, which we know the picture from a gas cylinder which will pass through, the column will be very long like a wounded, an injection spot where the gas will be the mobile phase and the stationary phase usually a liquid, and which will be GLC, and the computer and the waste compound can be formed because it's a heat is also generated there. It's a very careful, and we have to do this gas chromatography, and gas flow pressure will be controlled, and then we can have a sampling spot and the injection spot, and there will be a guard purification, and then the column, original column and detector, and then computerized, and the waste will be collected separately. So this is the instrument, actually original instrument, and we can see what is its gas is well established analytical technique, commonly for characterization, quantification or quantization, and you can say identification of volatile compound, usually gas chromatography. It can be used mainly in different fields such as pharmaceuticals, cosmetics, and even environmental toxins. And since the samples have to be volatile, human breath, blood, saliva, and other secretion containing large amount of organic volatile substances are easily analyzed using gas chromatography. So the powerful separation efficacy and the sensitization detection of gas chromatography is useful tool for the analysis of an essential oil. And despite of advantages, gas chromatography analysis in herbal products are usually limited in essential oils, such as possible degradation may be thermolabile and compounds, and requirements of volatile compound makes the gas chromatography unsuitable for many herbal compounds. You can write as a disadvantage. Available method: gas chromatography, gas liquid chromatography is there, gas solid chromatography is also there. So solid will be the stationary phase like silica gel or alumina or cellulose, which can be a stationary phase. Liquid, we know whatever the solvent, whichever you need, you can make it as an impregnated, maybe a water as a stationary phase, or any polar substance, a non-polar or lightly polar solvents can also be impregnated with the stationary phase, and it can be used as a column. And super fluid chromatography is already they have asked in 2 mark also separately what is super fluid chromatography. That is liquid carbon dioxide. It is an instrument, very sophisticated instrument. You can have a liquid carbon dioxide which will be passing through a pump will be there, and a modified pump, and then the pulse will be removed, any other than the any bubbles will be removed, and mixing column, and then analytical injection will be made into an oven where the column is there, through which the gas will be passed through, and detector, UV detector will be there, and then the waste will be collected, and it will be electronic backup will be there by means of a computer. So this super critical fluid chromatography is a hybrid gas of liquid chromatography which combines some of the best features in each, and this super fluid critical or fluid chromatography permits the separation to determine the group of compound that are not conventionally handled in the gas or a liquid chromatography. Liquid carbon dioxide is used, and it is applied for the separation of variety of materials such as drug, herbals, pesticides, natural and food products. And the super fluid chromatography techniques is especially useful for separation of any type of dimethyl polysilane or oligomers can be used, and separation of the compound molecular weight 400 to 700 deltas can be done by using carbon dioxide as a mobile phase, liquid carbon dioxide. So you should write liquid carbon dioxide. A person writes about the supercritical fluid which you are using, then that is the right answer for writing that polyaromatic hydrocarbon can be easily separated and supercritical fluid chromatography. So the second question for essay question which they have asked in December 2023, define and classify glycosides with examples. Write the source, chemical constituents and the method of extraction of senna. Very interesting. You have to write about the various classification of glycosides, examples, and then write about detailedly with diagrams of macroscopy, microscopy and the constituents and how you are extracting senna. Even if you can write about the oxalate apparatus or normal maceration technique, whatever you can write. But I have explained some of the very few points and the detail about the drug. You can just have glycosides definition. It is necessary for 10 mark question. In natural, glycosides are formed by interaction of nucleotide glycosides like uridine phosphate glucose with alcohol, phenol, steroid and terpenoid and flavonoid, etc. The joint glycoside linkages. That's why you are having A-glycan part. These are non-reducing, do not reduce by filling solution reaction, and organic substance which on hydrolysis yields one or more sugar molecules sometimes along with non-sugar molecule. There will be sugar molecule and non-sugar molecule. The sugar molecules are known as glycan and non-sugar molecules are known as A-glycan. Sugars are hemiacetal and occurs in an oxide ring, or you can say lactone ring. O will be there. So classification of glycosides which they ask, glycosides are generally classified in two ways. One is on the basis of their linkage. When you see O glycosides, sugar molecules, how it is joined with an A-glycan molecule. In O means, for example, with phenol OH group A-glycan, example amygdalin, endesin, arbutin, salicin, cardiac glycoside, androgen glycosides like senosides. Example you have to write. That's why they asked in the question paper very clearly. So students should know about the examples. N-glycosides, sugar molecules combined in nitrogen, N, and NH amino group of an A-glycan, nucleotides example. And S-glycosides, sugar molecules in combination with sulfur, SH thiol group, and that is A-glycan. You can say synergerin example. And C-glycoside, glucose molecule is directly attached with carbon atom, C atom of A-glycan. Example, androgen glycosides like aloin, barbaloin, cascaricides and flavon glycosides, etc. And you can write B, that is second one, on the basis of chemical structure of A-glycan or pharmacological activity, both together. So number one, androgen glycosides. These are derived from androquinones, possess purgative property, pharmacological property. Maybe dihydroxyphenol, example, chrysophenol, trihydroxyphenol amidine or tetrahydroxyphenol. That is androgen derivatives, often orange-red colored compound, soluble in hot water and alcohol. Example, sinucide A, B, C and D. So they have asked the 10 mark question, so you can write very clearly sinucide A, sinucide B, sinucide C and D. They are androgen glycosides. Then cardiac glycosides, you can write buffonolides, cardinolides, that also can be explained. Cardiac glycosides are composed of steroidal A-glycan with 5-membered ring structure, lactone ring, and acts on the cardiac muscle. These are also known as cardiotonic glycosides and cardiac activity depending on both A-glycan and sugar molecule. Activity resides in the A-glycan, but the sugar renders the compound more soluble, which increases the power of fixation of the glycoside to the heart muscle. These are divided in two groups, as I told, cardinolites and buffonolites. When you say cardinolites, they have 5-membered lactone ring in the C17. When we talk about the steroidal ring structure, phenanthrene, 3 6-membered ring with 1 5-membered ring and a 17th position. In the 17th carbon atom, you should know about cholesterol structure, steroid structure. These all should come in your mind. Very simple thinking in the imaginary picture should be there. In C17, if there is 5-membered lactone ring, then it is called cardinolites. Example, digoxin, that is digitoxin or olandrin. These all can be given as an example. Buffonolites, you see, buffonolites will have 6-membered instead of 5-membered, 6-membered lactone ring in the carbon 17 position. Steroidal ring. So example, silarinine A, which is in strophanthin and obtained from the squill also. Silarinine A is in strophanthin is in strophanthus, but silarinine A in squill. So this is buffonolites, cardinolites, a student can explain. And number 3, saponin glycosides, Dioscorea, they will be having a network like a form like many members of 6-membered ring structure will be there. These forms a honeycomb like structure, form, and shaken with water also they will make form and causes a hemolysis of the blood, and saponin complex organic compounds distribute in higher plants like Dioscorea are toxic to cold-blooded animals and lower organisms like earthworm and fishes. Depending on the aglycon moiety, saponins are of 2 types. One is steroidal saponin, tetracyclic triterpenoids. These contain cyclopentano, that means phenanthro, that means pentano perhydro phenanthrene ring, 3 6-membered ring, and diosgenin obtained from Dioscorea species, and pentacyclic, penta 5, that means triterpene saponin. It can be called a triterpene, terpenoids which have isopentyl pyrophosphate or dimethyl allyl pyrophosphate, which you might have studied about the 5-membered ring structure of a terpene, basic and isoprene units which is needed to make this pentacyclic. These are polymer of isoprene and mainly derived from beta amyrin. Example of glyceritic acid obtained from Glycyrrhiza glabra, Adimadira. So these all are some of the examples of saponin. And the next one is cyanophoric or cyanogenetic glycosides, the glycosides which are hydrolysis yield HCN, cyano drugs, which is that is benzaldehyde and sugars. Amygdalin obtained from bitter almond as an example, that is Prunus amygdalus, or prunicin obtained from wild cherry bark, you can write example. And isothiocyanate glycoside, these are sulfur containing compounds rich in and the family cruciferae, also known as glucinolates, and on hydrolysis yield isothiocyanate NCN group. So it can be written very clearly. And example, synergin for the bark and the black mustard, and synalbin from white mustard, and gluconapin from rapeseed. These all are the example of the 5th one. Bitter glycosides. These are complex organic compounds containing lactone ring, soluble in water and very bitter taste, and it must be diluted with solution for digestion, appetite, and gentiopicrin and amyrogentin, which are gentian root, and chirata. These all are you should write an example. And you can see coumarin glycoside. These are aromatic compound containing benzo alpha pyrone ring system. Example of these coumarin glycosides are asculin from the plant family Rosaceae and scopoline, And the flavonoid glycosides, eighth one, these are complex organic compound containing phenyl benzopyrone ring system. Example, rutin and quercetin and hyperside and diosmin in bouchi leaf, and hesperidin very commonly, which is present in lemon and orange peel, hesperidin. So these are flavonoid glycosides. So this is about the classification. You got to write about very clearly linkage OCNS, and also by means of pharmacology and their chemical classification, the 8 classification with example. And when we come to Senna, we know that you have done in the lab also, so it's easy for you to write about the Senna source. Senna is an example for androgen glycoside, and synonyms: Senna leaf, Tirunelveli Senna, Alexandrian Senna, African Senna, Cassia Senna, Indian Senna. And biological or botanical source, it consists of dried leaves of Cassia angustifolia and Cassia senna Wehel and Tirunelveli Senna, and Cassia acutifolia Delhi, that is also little leafy, will be round in shape, little not very, and Delhi it is Alexandrian Senna, and family Leguminaceae. So Cassia angustifolia, this is the flower which is having even having anti-diabetic effect, and Cassia acoustifolia flower will be little different, darker yellow, which I have shown in the picture. Geographical source, this Cassia angustifolia Indian Senna cultivated in Tirunelveli, Madurai, Tamil Nadu, Ramanathan district, and Andhra Pradesh, Gujarat, Rajasthan, and Cassia acutifolia Alexandrian Senna is indigenous to tropical Africa and is cultivated in Sudan and middle of upper Nile territory. So in the bank of the Nile river, usually the Senna is used as a cathartic, the higher version of even purgative, laxative and cathartic, laxative, purgative, cathartic for ancient times, and it is also included in Unani system of medicine, Senna. Highly medicinal use value, and nearly about 26 species are well known when contain anthracene derivatives is free from the glycoside state. And you can see one more point, among them Cassia angustifolia and Cassia acutifolia are in official monograph of pharmacopoeias, that we should know. And cultivation, they are available abundantly for the cultivation of Senna, 25,000 hectares, and land used in India were produced 22,000 of tons of leaves and 72,000 of tons of leaves per year, and they are a small shrub and having 1.5 meter height and containing compound of paraffinate leaf, and generally they are used in red loamy soil and coarse gravelly soil. They require better cultivation, but it can be grown in alluvial loamy soil, and the land is plowed and twice before sowing the seeds, and sowing done and broadcasting method, and fast growing should be done in February and or March, whereas second shown can be done in October or November. And seeds are having hard and tough and coating, and they are in pods and which are abiding in the surface and near to the induced quick germination and occur. Pore seeds are about sand mortar, and you can see from the pods, and 20 to 25 kilogram per acre seeds are required for cultivation. The plants are irrigated, favors light irrigation is only needed, and water logging and heavy irrigation should be avoided usually for this plant. It is sunny loving crop actually, and they grow early summer and February to March, or a winter in October and November. And Leguminaceae family, leguminaceous fertilizers can be used, applied for time to time. The plants are allowed to grow for about 5 months till flower stalk get developed, and the harvesting is done around after 2 to 3 months, and 3 stages. In the first stage, the leaves are plucked, and out of the leaves, leaflets, and thick they are found out, and quick grown and green in color. And the second plucking should be done after 1 month, and third plucking, the third one, should be done 4 to 6 weeks. And after third plucking, the plants will be uprooted. The plants will be uprooted, and the cenocidal content maximum of the leaf will be fully grown. So it is very good observed that the senna yield good. It is a cultivated crop, and maybe cultivation will be very good, and quality you can get, and dried in the shade, and it will be, and then 10 to 20 days and turns to yellowish green in color, and the leaves are separated from the pack and the bales and vacuum and stored in a dark and cool place. And you can see the leaves can be stored in for 5 years, because it is does not loses its potency. And this is the cultivation one video which I want to show how in North India and even in the Madurai Tirunelveli sites they are doing the cultivation in a manner, and how you can see it is ready for collection. You can see this is the area of hectares they used to use for the pots have been formed, and it was ready for collection, and extraction can be done for their isolation of cenocytes. See the view of how the cultivation, and interesting to see and it will be collected, and the drying area and containers will be there, and the greenhouses will be used for drying, and then everything will be dried in such a way and stored, and this is the way how they prefer for using the collection and the preparation. Macroscopic character: color is yellow green, odor is light, taste is miscellaneous and bitter. Size is usually the leaf will be 20 to 60 millimeter in length and 7 to 8 millimeter in width, and the shape of the leaf is lanceolate. Entire apex is acute, and spine in the top. See the leaves, dried one. And other characteristics are a small perennial under a shrub below 1 meter in height, and with ascending branches which you have seen the flowers, and bright yellow in color. And the full grown leaflet limits characteristic smell when crushed. Flowers are not season bound, and then grown between 35 to 70 days of age. And other species are very similar of Cassia angustifolia in morphology, and the growth are expected to dimension of the leaf, and the pods which are shorter and narrow. There are many other species. This is the schematic diagram of the section of a leaf, midrib, how the fiber and upper epidermis and lower epidermis, the palisade cells will be, upper palisade, lower palisade will be different from other leaf. When you take a datura or any type of leaf and you take, it will not have this upper palisade lengthy lines and vascular bundles, and the xylem how it is, and the colonchima cells in the lower midrib will be seen very interestingly being iso-bilateral. That is why it is called iso-bilateral leaf. You should specify this. And senna shows more or less similar features of both surface of the leaf and few differences. And transverse section of leaf shows upper and lower epidermis and straighted wall cell, few of which contain mucilage. And you can see the pictures with, you can write covering trichomes and upper epidermis, and clearly you should draw a diagram with pencil, and lower epidermis and with the inner xylem vessels, and as well as the colonchima cells in the lower midrib region, and lower stomata can also be shown, and saprophytes and the colonchima presence of, and you can say that the parasitic stomata, that is parasitic stomata, and non-lignified uncellular trichomes are found in the both surfaces up and down, and the single layer of palisade parenchyma observed in the both sides, upper and lower, but and discontinued in the midrib region, in the lower region. Lower epidermis, you can see in the palisade long cells will be discontinued in the lower midrib region where the colonchema or the colonchematous tissues are drawn. You have to draw very clearly. The palisade is followed by spongy mucilage containing cluster of calcium oxalate crystals, that is what saprophytes, which is shown. Midrib shows a vascular bundle of xylem and phloem, and most surrounded by the lignified pericyclic fibers and sheets of parenchyma which contain prismatic, that is why saprophytes, prismatic calcium oxalate crystals are there. And senna leaf powder microscopy, when you see a powder of you can see epidermis, polygonal cells, unicellular thick walled trichomes can be seen in the powder, and you can see the unicellular palilose non-glandular hairs, that means trichomes, and the long palisade cells, lower and upper under palisade cell, single row of palisade layers, and stomata, which you can see that is parasitic stomata in both the surfaces, and powder microscopy also, which we can see. Senna leaf powder can also be seen very clearly, the clear clat that is an axial surface, the cluster of crystals of calcium oxalate saprophytes throughout the lacnose powder can also be seen, and crystal sheath as shown, a glitter shaped group of similar fiber on the apaxal side of prism crystals of calcium oxalate can also be seen. Chemical constant, we know that senna contains senoside A and B based on the A glycon, senoside A, B, senoside C, D, glycoside hetero androquinones, are heterodiandrones of alloamodin and ruin present, or rutin may be present, and other palmidin A or ruin andron and alloamodin glycosides are also present. Senna also contain free chrysophenol and amodin, their glycosides, and free alloamodin ruin, their monoandrones, diandrones, and other glycosides. Mucilage is present in the epidermis of the leaf and gives a red color in ruthenium red. This is a structure that is a R, we can see that is C6H12O6 and C6H11O6, here also C6H11, that is a linkage of O glycoside glucose molecule with the phenanthrene, or you can see 3-membered and ring structure of 6-membered, how R can be changed by acid or alcohol in meso, trans, and form of two stereochemical form of senoside A, B, and C, D can be easily mentioned with one structure which you study. And this is about the senoside A and B. Again, I have written about the glucose molecule to show you how the C12 and H6, how it is made into a lactone ring structure in a glycoside linkage, which is shown for both the senoside A and C, and D and E, how they are isomers, how they are geometrical isomeric structures can also be drawn by you. Silica, that is senna leaf, are containing anthraquin glycosides a small amount, and they are C and D, ruin D glycosides, and diglycoside and alloamodin, and containing phytosterol, mucilage, resins, and miceryl alcohol, and chrysophonic acid and salicylic acid. Chemical test, bone tracker test. You have to write about bone tracker test. There is a modified bone tracker test by using ferric chloride. Drug is boiled in sulfuric acid and filtered, and the filter is added with benzene or ether or chloroform and shake well. And separated organic layer, when you show with ammonia, pink color or red color will shows the ammonical layer confirms the presence of anthraquin glycosides. So uses, senna leaves are used as a laxative, and it is cause an irritation to the large intestine and have some gripping effect. Thus they are prescribed along with the uh, with the that is carbamates. Senna is stimulating agent, cathartic, and exerts an action of increasing smooth muscle and large intestine. And the marketing products are also available, marketed product like Softverb in Lupin Laboratories, and Loswa in Kalbab tablets are also available in health sector, and the Kultab tablets and marketed. And elite drugs, Senna, Poda, you got right, and Podacarpa, Senna, Auriculata, and Senna, and Cassia, Auriculata, Cassia, Podacarpa, and Cassia, Abovata or Doxenna. Difference, there is difference between them. You can see that is it is seldom of 40 centimeter Alexander Senna, very seldom of 50 centimeter length, greyish green and yellowish green, and more asymmetric in the base and less asymmetric in the base, and more broken in the curved breads, and this is seldom broken, usually flat. And stomata is about 10 to 15 usually, and here 14 to 20. And this one vein eyelet number, when you see in camera lucida, 25 to 29.5, and this is 19.5 to 22.5. And this is the leaf, Alexander Senna and Port's picture, Tirunelveli Senna. Difference between which I have told, even you have to add in TLC test, pink and daylight and orange light, and also stomata, hair, numerous like trichomes, and 6-hydroxy muslin, and these are the differences. And I did not add in the answer, they were asked isolation. Isolation I will discuss in the class. I will give you one more, how to isolate. You write about the maceration technique, extraction technique by means of Soxhlet apparatus, and isolating this compound can also be added. 10-mark question is very important. They have asked in the question, especially how you are extracting senocytes, and senocyte A and B, whatever you are explained, you can write a small explanation about extraction by means of Soxhlet in small quantity or in large quantity. We can go for using steam. You can use very good chambers for boiling the leaves and extracting, and we can filter it, and also that can also be maceration techniques can also be followed. Anyhow, there is notes, there is another chapter we have separately, how will you isolate senocytes, they have asked, and this is 10-mark question. This much itself you will come 3-4 pages it is needed. And so that we have finished with the 2 essay question, and we will continue in the next class about the short question. We have 7 short notes, 5-mark question, 10 brief 2-mark question also have to be discussed. So that my students, I request you to write this 2 essay question clearly, and in a Senna especially you add little more points in the chapter. How will you isolate senocytes from the leaves or in the pods of the plants after cultivation, after collection? Because the 10-mark question, if you add a few lines about isolation of and the bone tracker test, and also writing about modified bone tracker test for C glycosides. This is O glycosides, some of them which is C glycosides, not for Senna. We can aloes, which we can also add. Thank you for listening. Write down, and you will be succeeding. Think positive and follow my instruction. You will never have a negative of failing any this subject. You will pass 100%. Thank you for listening.